Good day, students. This is Dr. Francis Ian Salaver, and I will be talking about antigens. There are two terms in immunology that are always used interchangeably. But since you are studying immunology right now, you learners should know the difference between the two. But first, let us know how the scientists coined the terms antigens and immunogens. Antigens because they are known to generate antibodies, antibody generators. Immunogens because they are known to generate immune responses, immune response generators. But what is now the difference between an immunogen and an antigen? Take note, an immunogen is any substance that when introduced to our body will cause the immune system to produce antibodies or activated T cells. Take a look at this bacterium. Can you appreciate that it has substances on its surface? Those substances will only be considered as immunogens if they are introduced into the immune system, and they will make the immune system generate antibodies specific against them. Antigen is a term that is used to refer to any substance that can bind to an antibody or to a T cell receptor. I want everybody to understand the antigen is not the substance responsible for the generation of the antibody. So what do I mean by that? So I will explain that on the succeeding slides. The blue triangles are antigens because they can bind to specific antibodies. That's how we define antigens, right? Any substance that can bind to an antibody will be referred to as antigen. But if the substance can make the immune system generate antibodies, you refer to it as immunogen. So what is now the difference between immunogen and antigen? So for you to understand that one, I want you to imagine that you are performing blood typing. So what you will do is you will place drops of blood on top of a slide. Then after that, you will add on each drop of blood anti-A sera and anti-B sera. Anti-A contains an antibodies directed against the A antigen present on the red cells, while anti-B contains antibodies against the B antigen present on the surface of the red cells. Let's assume that the blood that you are processing for blood typing was derived from someone who is blood type B. So expect that on the slide or the test tube that contains the RBCs, the red cells added with anti-B sera will agglutinate. It's because the antibodies attach themselves to the B antigens on the red cells, cross-linking now one red cell to the other red cells, resulting to agglutination. In this case, the B antigen on the surface of the red cells of the person is an antigen. Why? Because we define antigen as a substance that can bind to an antibody. But I want everybody to understand that in this case, the B antigen is not acting as an immunogen, because the definition of immunogen is that it should be the chemical or substance responsible for the production of the antibody. Do you know that the chemicals we have in anti-A and anti-B seras are derived from plants? Anti-A sera was derived from the plant Vicia craca, while anti-B sera was derived from Griffonia simplicifolia. So what I want everybody to understand, the B antigen on the red cell of the person is not the reason why you have the anti-B sera. What the B antigen did is just to bind to the antibodies. So therefore, in this case, the B antigen on the red cell of the patient is just acting as an antigen. I want everybody to understand, since immunogens can induce the production of antibodies by the immune system, and antibodies tend to be specific, expect that the immunogens will also bind to the antibodies produced because of them. So, immunogens can also act as antigens because they can bind to the antibodies. Take a look at this virus and look at the substances present on its surfaces. These substances are immunogens because when they were introduced to the immune system produced antibodies. These antibodies are specific to the substances that induced their production. So expect that the substances present on the virus will bind to the antibodies produced against them. So in this case, the substances present on the surface of the virus are also acting as antigens. So what can we conclude from this one? Immunogens, aside from initiating antibody production, can also act as antigens because they can bind to the antibodies produced against them. But antigens, okay, will never act as immunogens because they can just simply bind to the antibodies specific to them. So therefore, we can conclude all immunogens are also at the same time antigens, but not all antigens are immunogens. There are substances, however, that when you present to the immune system will not generate any immune response, primarily because they have low molecular weight, too small to be taken seriously. And since they cannot induce the immune system to generate immune response, this group of chemicals are referred to as incomplete antigens. Incomplete antigens are known as haptens. That's why in the third bullet, they are described to be substances not immunogenic by themselves. But there's a plot twist, and I will mention that later on. Again, an immunogen is a substance that when introduced to the immune system will cause the immune system to produce antibodies, and these immunogens will also act as antigens because they can bind to the specific antibodies produced against them. A hapten or an incomplete antigen when introduced to the immune system will not create or generate an immune response. For you to understand the importance of studying haptens, I want you to imagine a little boy who is always bullied at school because of his small size. But if that little boy will bring his dad at school, the boy will be able to fight off his bullies. So how is that related to the discussion of the hapten? I want everybody to take a look at the left side of the photo. Haptens have small molecular weight, thus they cannot induce the production of immune response. But if the hapten will bind to a carrier, the green colored figure in the picture, so if that carrier will bring along with it on its surface the hapten now will gain the size to become immunogenic. So that's why if the hapten is bound to a carrier or a protein, it will now become immunogenic. This is the hapten and this is the carrier. And since the hapten is now attached to the carrier, the hapten is now considered as immunogenic. If the carrier and the hapten will be introduced into the immune system will produce three types of antibodies. What are these? The antibodies directed to the hapten, the antibodies directed to the carrier, and there's a third group of antibodies that can bind to both the hapten and the carrier. So, the important point that I want you to remember is once the hapten is bound to a carrier, the immune system will attack both the hapten and the carrier. Hapten becomes immunogenic if it is bound to a carrier. If both of them are introduced into the human body, the immune system will generate antibodies against the hapten, antibodies against the carrier protein, and antibodies that can bind to both hapten and carrier proteins. Methyl Dopa is one of the antihypertensive medications. It has low molecular weight, thus it will never make the immune system generate immune responses against it. The problem with methyl dopa is that they have affinity to the membrane of red cells. And why do you think it's important to take note of that? Methyl dopa, since it is a hapten, if introduced into the immune system, it will not generate any immune response. But the methyl dopa can coat itself around the red cells. The red cell now will serve as a carrier, giving now the methyl dopa the size enough for it to become immunogenic. And let's apply what you have learned. If the carrier and the hapten will stimulate the immune system will generate antibodies against the hapten, methyl dopa, against the red cells, and antibodies that can bind to both red cells and the methyl dopa. So, in some people who are taking methyl dopa, they will develop what we call drug-induced hemolytic anemia. The immune system was made to produce antibodies that will attack the red cells of that person. So how to manage this case? Withdraw the drug. Stop the administration. The next term that I want you to learn is the term epitope. Epitope is also known as antigenic determinant. Please take note of the definition of epitope. It is the site in an antigen where the antibodies will specifically bind onto. So take a look at the picture of the antigen on the PowerPoint slide. The area where the antibody will bind to that antigen, you call that as the epitope. And the corresponding site on the antibody is called paratope. Aside from defining epitope as the site where the antibody will bind, it's also very important to take note that it is that portion of the antigen that stimulated the immune system to generate antibodies. That's why of all the sites present in the antigen, the antibody is directed to that particular area, and that's the epitope. I want everybody to understand that for each microorganism, you could have multiple epitopes. So therefore, every time you will have an infection with a particular bacterium, you may generate more than one type of antibodies. And the antibodies you generate against the bacterium will always be specific to the epitope that induced their production. So in the picture, the triangular shaped epitopes are the ones specific to the IgM because they are the ones that induce the production of IgM. But expect that the other epitopes in the picture will also have their own corresponding antibodies. There are cases that microorganisms may share the same type of epitope. And what will happen is antibodies produced against organism number 1 will also attack organism number 2 because they share the same types of epitopes. And you call that phenomenon as cross-reactivity. So I'll give you examples of applications of cross-reactivity phenomenon. The discovery of what we call today as vaccination was because of cross-reactivity. Thanks to Edward Jenner. I hope we can recall in the history of immunology, Edward Jenner inoculated cowpox secretions into the skin of a young boy. The young boy developed the disease. But since cowpox is a mild disease, it is a self-limiting disease, the boy eventually recovered. And what Edward Jenner observed was the kid never contacted smallpox because the viruses causing smallpox and cowpox share the same epitopes. So that's why the antibodies generated against cowpox virus also attacked smallpox virus because of cross-reactivity. I know you have also heard in the past that if you will have recurrent tonsillitis, it's better to have it removed by surgery because recurrent tonsillitis will eventually involve now the heart valve. So that's the complication. I want everybody to take note that the causative agent of streptococcal pharyngitis is Streptococcus pyogenes. Shown on the right side of the screen is one Streptococcus pyogenes bacterium, and please appreciate that one of the antigens present in its wall is the M protein. That M protein, I want you to look for it first. That M protein allows the Streptococcus pyogenes to escape phagocytosis. If you will be infected with Streptococcus pyogenes because you're having sore throat, expect that one of the antibodies that you will produce is antibody to M protein. And since antibodies fall under adaptive immunity, it would only mean that the more you will be exposed, the higher amount of antibodies that you will generate. If you will have recurrent tonsillitis, expect that you will be again re-exposed to M protein. Expect that you will produce higher amount of antibodies. Until such time that the number of antibodies that you have in the blood can now also act on the antigens present on the heart valves. And those antigens have the same morphology and appearance as that of the M protein. That's why the antibodies in your blood thought that they were the M proteins of Streptococcus pyogenes. So these antibodies will attack your heart valves, resulting now to what we call rheumatic heart disease. So rheumatic heart disease is an example of what? A cross-reactivity phenomenon. There are several ways on how to classify epitopes. First of which is to classify how many types of epitopes are there and how many of these epitopes are present on the surface of the organism or cell. So, when you say determinant, this is referring to the number of types. When you say valent, how many of these types of epitopes are present on the cell. So, when you say unideterminant, one type, univalent, only one of that type of antigen is present on the surface of the cell or microorganism. So, take a look at these red-colored cells here. Can you appreciate that they are displaying A antigen? So, there's only one type of epitope. You will use the term unideterminant. And for each cell, you only see one A antigen. So, there's only one valency. So, you will use the term unideterminant, univalent antigen. How about unideterminant, multivalent? When you say unideterminant, there's only one type of epitope, but you have many of these epitopes on the surface of the cell because you use the term multivalent. So take a look at one cell. Again, the cell only has A antigen, but you have several of these epitopes surrounding the cell. Thus, unideterminant, multivalent. How about multideterminant, univalent? So when you say multideterminant, there should be several types of epitopes, but for each epitope, you should only see one of it on the surface of the cell or microorganism. So take a look at one of the cells in the picture. As you can see, it has three types of epitopes, A, B, C. Thus, you will use the term multideterminant, but you only see one A, one B, and one C epitopes, right? So that's why you will use the term univalent. So when you say multideterminant, multivalent, there should be several types of epitopes and many of each type of epitope on the surface of the cell or microorganism. So take a look at the picture of the bacterium. It has triangular epitope, it has cube epitope, it has spherical epitope. So you have a total of 3 epitopes. And for each epitope, you have more than one kind. You have several triangles, you have several cubes, and you have several spherical shaped epitopes. So therefore, that bacterium is described as multideterminant, multivalent. Epitopes can also be classified as linear or conformational epitopes. Majority of the epitopes are proteins by nature, and proteins are made up of amino acids linked together by peptide bonds. So I want you to take a look at the red arrow in the picture that is pertaining to the cell wall or cell membrane of the bacterium. and then displayed on the cell wall or cell membrane of the bacterium are the different epitopes. This is an example of a linear epitope. When you say linear epitope, it is composed of sequential series of amino acids. So, those green colored amino acids there will serve or form one epitope, and they will be recognized by the specific antibody against them. However, there are what we call conformational or discontinuous epitopes, wherein they are actually produced by folding in the proteins. Can you appreciate that this type of epitope, the conformational, is formed when the protein folded, and the fold in the protein brought into closer proximity non-sequential amino acids, the ones highlighted in red. They are not sequential. They are from different segments in the protein, but they are brought to closer proximity because of the folding. And what will happen is those non-sequential amino acids will now form one epitope, which will be recognized by a specific antibody. Let's have a review of the structures of proteins that you discussed in your biochem. If you only see series of amino acids linked together by the strong peptide bonds, then you are looking at primary structure. But if the protein will start to have folds in it, it can form the secondary and tertiary structures. If one protein with tertiary structure is made to combine with another protein with tertiary structure, that's the time you get to form the quaternary structure of proteins. I want everybody to understand that peptide bonds are the only ones that are considered to be strong bonds in the bonds that you have here in the different structures of proteins. So therefore, the hydrogen bonds, the hydrophobic bonds, the electrostatic bonds present on secondary, tertiary, quaternary bonds are weak bonds. Therefore, if you subject these proteins to denaturation, the quaternary, tertiary, secondary structures will all be lost. But since the primary structure is formed by the strong peptide bonds, the primary structure is not prone to denaturation. And I want everybody to take note that the linear epitope is most of the time appreciable in the primary structure because linear epitope is composed of sequential amino acids, which you can clearly see in the primary structure. And since conformational epitopes are formed when proteins fold, most likely they can be formed in secondary, tertiary, and quaternary structure of proteins. Linear or sequential epitopes are composed of sequential amino acids that can be recognized by antibodies. Conformational or discontinuous epitopes consist of residues that are discontinuous in the proteins that were just brought in close proximity because of the folding of the proteins as it formed its secondary, tertiary, or quaternary structures. So what is now the importance of memorizing that there is linear epitope and there's also conformational epitope? Shown on the left side of the screen is a folded protein. So expect that you have there the weak bonds that will form these secondary and tertiary structures of proteins. If that folded protein will be denatured, all of those bonds will be lost. What will now be retained is the strong peptide bond. That's why, as you can see in the unfolded protein, you only see there the primary structure of proteins. Now take a look at this linear epitope. It is composed of sequential series of amino acids, and that linear epitope is recognized by these antibodies. If the folded protein will be denatured, the sequence of amino acids will never be changed. That's why even if the protein was already denatured, the antibody can still look for that sequential series of amino acids and bind to it. So what do I mean by that? Linear epitopes, even if they are subjected to denaturation, will still be recognized by the antibodies Because the sequential series of amino acids are linked by peptide bonds, and peptide bonds are very strong. That's why the linear epitope will remain the same, and it will still be recognized by the antibody after denaturation. In the case of conformational epitopes, they are formed when proteins form folds, and the folds will bring to closer proximity certain areas or certain amino acids, and these non-sequential amino acids will now form the epitope. So that conformational epitope will be recognized by the specific antibody. If that protein will be subjected to denaturation, all of the folds will disappear. And what will happen? The conformational epitope will be divided into several segments. And the antibody will no longer be able to identify the epitope. The antibody would say, "Where is it? I cannot find it anymore." So, conformational epitopes, if subjected to denaturation, will no longer be recognized by the antibodies or sensitized or activated T cells. Epitopes can also be classified as accessible or inaccessible. The blue-colored epitope is accessible, while the pink-colored epitope is inaccessible because it is found inside the microorganism. You know very well that B cells have surface immunoglobulins, and they are using these immunoglobulins to recognize specific antigens so that they can present the antigens to T helper cell. And please recall in our discussion that there are two types of antibodies present on the surface of B cells. You have IgM and IgD. What is used by the B cell to recognize its specific epitope is IgD. So I want you to assume that the black-colored Y-shaped figures surrounding the B cells or displayed on the surface of B cells are IgD. Now, between accessible and inaccessible epitopes, B cells can only recognize accessible epitopes because their surface immunoglobulins do not have means of identifying the epitope that is hidden inside the microorganism. T cells, on the other hand, can recognize both accessible and inaccessible epitopes. Why? T cells do not have surface immunoglobulins. So how can they recognize both accessible and even the inaccessible epitope? So please take a look at the red-colored circle. It has blue-colored accessible epitope and pink-colored inaccessible epitope. If that microorganism, red-colored circle, will be engulfed by the blue-colored macrophage or antigen-presenting cell, the antigen-presenting cell might sample both the accessible and inaccessible epitopes and load them in MHC class 2 and present them on their surfaces. And you know very well that if the antigen is presented using class 2 or class 1 MHC, they will now be recognized by T cells. So T cells can recognize both accessible and inaccessible epitopes provided that they are first processed and presented by an antigen presenting cell, because T cells themselves cannot directly recognize the epitopes. They need to wait for the macrophage to choose these epitopes during antigen processing and display them using MHC class 1 and class 2. So, let's review. Without help, the B cells can use its surface immunoglobulin to identify, recognize accessible epitopes, but they cannot recognize inaccessible epitopes. T cells, in contrast to B cells, can identify both, but the epitope should be first presented by an antigen-presenting cell. So let's have a short quiz. B cells can recognize, yes, accessible epitopes using its IgD surface immunoglobulin, while T cells can recognize, yes, both accessible and inaccessible epitopes, provided that they are presented by an APC. We have these sets of personality and physical traits that we are looking for in a guy or a girl before we will consider them as our ideal person. The immune system also has its own list of traits that it will look for in a substance before it will consider the substance as immunogenic. What are these traits? A substance must have all of these characteristics before it will make the immune system generate an immune response against it. So what are these characteristics? The substance must be foreign. The substance must be degradable. The substance must have high molecular weight. The substance must be structurally stable. This is in contrast with number 2, no? How can something be structurally stable but degradable? I will explain that later on. And the substance must have chemical complexity. Let's start with the first one, foreignness. A substance must be considered as non-self or foreign for it to induce an immune response. So foreignness is the degree to which an epitope or antigenic determinant is recognized as non-self by the immune system. So how will the immune system say that this is foreign and this is not? Any substance that never came in contact with the lymphocytes during the embryonic period will be considered as foreign. Why do you think it's very important to take note that a substance will be considered as foreign if it never came in contact with the lymphocytes during their maturation? Can you remember the negative selection of T cells happening in the medulla of the thymus? In the negative selection, the single positive T cells were introduced with self-antigens, and they are expected not to react because if they will react, they will be programmed to apoptosis. See, during the maturation of T cells and B cells, they are already taught what is self and what is non-self. And that is called immunologic tolerance. They should tolerate the presence of self-antigens. Don't worry, we will have a separate chapter on immunologic tolerance in our final period. Another important characteristic of an immunogen is that it should be structurally stable. For me to explain what do I mean by structurally stable, I want you to imagine an antigen that is introduced to oral route. Once swallowed, the antigen will lodge in the stomach, and the stomach is known to contain hydrochloric acid. If the antigen is structurally stable, once it will reach the stomach or maybe the small intestine, and you have their hydrochloric acid and enzymes, that substance must be able to resist degradation. Why? Because the substance should be absorbed in the stomach and intestine and should come in contact with the macrophage so that the macrophage will sample parts of its epitopes to T cell. Because if that's the case, then the chemical is becoming immunogenic because it was able to activate the T cell, and the T cell will make the B cells become plasma cells and produce antibodies. A substance is not structurally stable if inside the stomach, the substance is already broken down and it is no longer absorbed in the stomach or intestine to be presented by the macrophage to T cell. And that's what I mean by structural stability. Now you have understood that for a substance to become immunogenic, it must be structurally stable. But the third property states that the substance must be degradable. What do we mean by this? Antigens that activate the T cell and later on the B cell to become plasma cells, and there will be production of antibodies afterwards, must be presented by the antigen presenting cell using the MHC molecules. And you know very well that the APC does not present the whole microorganism, does not present the whole cell. The APC must use the lysosomes to break down the antigen into small pieces, and choose what part is the most immunogenic and present that particular part using the MHC on the surface to activate the T cell. When you say degradable, the substance must be easily degraded by the macrophage for antigen processing and presentation to proceed. A substance is structurally stable if it can withstand the hydrochloric acid in the stomach, the enzymes in the intestine, and then it will be absorbed in the body, it will go to the blood and encounter the monocytes and macrophages, which are our antigen-presenting cells. But once inside the antigen-presenting cells, the substance must be readily degradable so that the macrophage can present a portion of that substance on its surface to activate the T helper, which will later activate the B cell. A substance is not immunogenic if it is not degradable because the macrophage will not have any way of presenting it to the T cell. And the T cell will not also activate the B cell to produce the antibodies. For a substance to be immunogenic, it must be foreign, it must be structurally stable, it must be readily degradable by the APCs. Number 4, it should have a high molecular weight or big size, because with big size, there's a chance that there could also be high numbers of epitopes. Take note, haptens usually have molecular weights of less than 10,000. That's why they are considered as incomplete antigens, because they cannot induce the immune system to create immune responses. Higher than 10,000, the substance will now start to become immunogenic. The last but not the least is that the substance must have chemical complexity. What do we mean by chemical complexity? So for you to understand what's chemical complexity, I want you to compare polysaccharides and proteins, both of which are immunogenic because they have huge sizes. But the thing is, why proteins are more immunogenic than polysaccharides? So I want you to imagine a polysaccharide. A polysaccharide is formed by long chains of monosaccharides. So if you have a polysaccharide, it appears to be long. But if you're going to take a look at its structure, you will only see 3 types of monosaccharides. So, for example, you have a long polysaccharide. Its size may be overwhelming, but when you look at it, you'll just simply say, okay, it's composed of glucose, galactose, and fructose, and another glucose, another fructose, another galactose. Oh my God, you're so predictable. So, the immune system will think, okay, you have less chemical complexity. In the case of proteins, proteins can be formed by 21 amino acids. So if you have, let's say, a protein that has the same length as the carbohydrates that we previously discussed, but if you're going to take a look at the protein, you will notice that it has valine, tyrosine, phenylalanine, isoleucine, leucine, cysteine, histidine. Diba? You will really look at it as chemically complex. So that's why proteins are more immunogenic than polysaccharides. Plus, aside from the fact that proteins are known to have high molecular weight. So, in terms of molecular weight, proteins are better than polysaccharides. In terms of chemical complexity, proteins are also better than polysaccharides. So, the most immunogenic substances are proteins. It's also very important to take note that the protein with the higher number of phenylalanine, tyrosine, and tryptophan will be more immunogenic than another protein which has less of this aromatic or non-polar amino acids. In terms of chemical complexity, proteins are more complex than polysaccharides, polysaccharides are more complex than lipids, and lipids are more complex than nucleic acids. So So let us now discuss the different types of antigens. When you say autologous antigens, the auto there would mean self. So these are the antigens that are found in your body. Take note, these are the antigens that came in contact with the lymphocytes during their maturation. That's why they are not considered as foreign by the body. If they will be considered as foreign due to some disorders, these autologous antigens can trigger a group of diseases we refer to as autoimmune disorders. And in autoimmune disorders, your immune system is generating immune responses against your own antigens. Not all of the self-antigens are present during the time that the T cells and B cells are undergoing maturation. Male babies do not have yet mature sperm cells. And when we were born, we still don't have mature cornea. So these antigens never came in contact with the lymphocytes during their maturation. So they can be considered as foreign. The body is so wise that the body will try to contain these antigens in areas that cannot be reached by the immune system, so that the immune system will not recognize them as foreign and the immune system will not attack them. And these types of antigens are called sequestered antigens. Since cornea is a sequestered antigen, one can donate the cornea to another person without matching them. Why? Because the cornea is found in that portion of the eye that doesn't have blood vessels. So even if you will place there a foreign cornea or a cornea from another person, the immune system will never have any means of attacking the cornea because, again, it is a sequestered antigen. This is the picture of the testis. You know very well this is where sperm cells are produced. Please appreciate the lobules inside the testis. And take a look at one lobule. You have there seminiferous tubules. This is how one of the tubules will appear under the microscope. I want you to imagine that on one seminiferous tubules, you have there developing sperm cells, and you also have their blue-colored cells, which are labeled as Sertoli cells. Please appreciate in the picture that the Sertoli cells are positioned near the base of the seminiferous tubule, where you can find the capillaries. The main purpose of the Sertoli cell is to filter the substances that will enter the seminiferous tubule. Why? Because some of the substances can cause the DNA in the developing sperm cells to mutate, and the Sertoli cells will not allow that one to happen. Aside from regulating the entry of substances into the seminiferous tubule, these Sertoli cells will provide what we call the blood-testis barrier, and that blood-testis barrier will not allow immune cells and other components of the immune system to enter the seminiferous tubules because sperm cells are sequestered antigens. They are only produced after, or they start to appear after puberty. So I want you to take a look again at this one seminiferous tubule. Please appreciate that this is the Sertoli cell. Another type of antigen is the isogenic or syngenic antigen. So from the name syngenic, meaning the persons have the same genes. So these are the antigens shared by people with the same genetic makeup, or basically twins. The next type of antigens, allogenic antigens. These are antigens shared by individuals of the same species, meaning we're going to compare a human to another human, Streptococcus to another Streptococcus bacterium, a cow to another cow, frog to another frog. But take note, these antigens are only present in some, but not all of the individuals of the species. For you to understand that, I will use an example of allogenic antigens in human, the ABO antigen. I am blood type A, and I also know other people who are blood type A, but not all of the human species have blood type A antigens. But then again, A antigen is something that is shared by individuals of the same species. Antigens that are shared by individuals of unrelated species or different species are called heterophile antigens. One good example, the M protein of Streptococcus pyogenes, the causative agent of pharyngeal tonsillitis, and the antigen of the human heart valves. That's why if you have recurrent tonsillitis caused by Streptococcus pyogenes, the antibodies against M protein will cross-react with the antigens present in our heart valves. Humans and bacteria are unrelated species. I'll give you another example of heterophile antigens. You have here the Epstein-Barr virus, the known causative agent of infectious mononucleosis and Burkitt's lymphoma. The person who develops the infection with Epstein-Barr virus are expected to develop antibodies against the virus. But there is that what we call as force-man antigen. And this antigen is shared by different types of viruses, including the Epstein-Barr virus, bacteria, plants, and animals. So meaning, the antibodies produced against Epstein-Barr virus, particularly its force-man antigen, may also bind to bacteria, plants, and animals. Do you know that the antibodies humans produced against Epstein-Barr virus can actually bind to sheep red blood cells because sheep red blood cells contain force-man antigen similar to Epstein-Barr virus. That's why in immunology serology laboratory, we identify the presence of antibodies against Epstein-Barr virus in the serum of the patient not by mixing the serum of the patient with the virus, but by mixing the serum of the patient with sheep red blood cells. If the sheep red blood cells will agglutinate, it means that the patient has the antibodies against force-man antigen shared by a virus, Epstein-Barr virus, and an animal, the sheep red blood cell. In this case, the force-man antigen is an example of heterophile antigen. So if the sheep red blood cells will agglutinate, that's your clue. The patient could be suffering from an infection caused by Epstein-Barr virus. Another type of antigen is exogenous antigens. These are antigens that we can find in the external environment and they will just enter our body through several ways. They can be the pollens and the microorganisms in the air. They can be the drugs and the vaccines that are injected into our body. So as long as they are from the external environment, you consider them as exogenous antigens. Some antigens are considered to be endogenous, meaning they are produced inside our bodies. Majority of these substances are products of cell metabolism. So every time the cell will produce a waste product, that waste product can be considered as endogenous antigens. Majority of the endogenous antigens have low molecular weight. That's why we do not basically produce immune responses against them. Good examples of endogenous immunogens or antigens are the substances that the cells in the body would produce if they are infected by viruses or intracellular bacteria. So take a look at these viruses in the picture. They are about to invade a human cell. Inside the human cell, these viruses will produce proteins. There's production now of viral proteins because the cell was infected by a virus. And you know what the cell will do? It will get some of the viral proteins, load them on MHC class 1, and present them on the surface so that the viral protein will be recognized by the T cytotoxic T cell. In this case, the viral protein was only produced when the cell was infected by the virus. So that viral protein is an example of an endogenous antigen. Take note, exogenous antigens, since they are found outside the body and they will just enter the body through several ways, they are considered to be extracellular antigens, they will be presented using class 2. Endogenous antigens are produced inside the cells because the causative agents producing them are found inside the cells. So, they are best presented using class 1 MHC. The next type of antigens that I want you to learn are the super antigens. These are, most of the time, bacterial exotoxins. When you say exotoxins, these are toxins produced by gram-positive bacteria. And do you know what's wrong with these exotoxins? They can stimulate the immune response, but the problem is the immune response is characterized by excessive T cell activation. So excessive that the activation of T cell can now cause tissue damage. Another important thing about super antigens is that they skip the usual antigen processing. Now, take a look at the picture. You know very well typical antigens are engulfed by the APC. They are degraded by the APC, and portions of them will be loaded on class 2 MHC. And once loaded to class 2 MHC, they will now be displayed on the surface. Right? That's what we mean by antigen processing. If the antigen now will be recognized by the T helper cell, then that's what we call as the antigen presentation. Antigen processing is usually skipped in cases of super antigens. What do we mean by that? Take a look at this picture. Please appreciate that we have the T cell on top. You have the antigen presenting cell below. You have here the blue colored figure representing MHC class 2. Then you have there the green colored figures representing the T cell receptor. The red colored figure is representing the antigen. That's the typical antigen. It's supposed to be found between the MHC class 2 and the T cell receptor. If the antigen is presented in this manner, the antigen will only be recognized by a specific T cell. So only the T cell that is specific to the antigen will be activated, and it's only those T cells that will activate B cells to produce the plasma cells and antibodies. The problem with super antigens is that they are not positioned between the MHC and the T cell receptor. Look at the picture. These super antigens will bind to both MHC and T cell receptor, and they will serve as a clamp. They will not allow the two cells to detach from each other. Please recall that after the T cells will be activated, they should detach. The antigen presenting cell and the T cell should detach because the T cell has to activate B cell, and the macrophage has to produce interleukin 1, interleukin 6, tumor necrosis factor. In the case of super antigens, the super antigen will not allow the dissociation between the APC and the T helper cell. So, the APC will constantly activate the T cell. And the T cell, in response to that, will produce large amounts of cytokines. And the problem now is large amounts of cytokines can be destructive to human tissues. Remember, if the antigen is presented in the middle of MHC and T cell receptor, only the specific T cell will respond to the antigen. In the case of super antigen, since it will just serve as a clamp, any macrophage can be bound to a T cell with T cell receptor. And since that would happen by random, these super antigens can lead to non-specific T cell activation. That's why expect a lot of T cells will be producing large amounts of cytokines. Because if it is just the typical antigen, only the specific T cell will produce the cytokines, and the level would not be enough to cause tissue damage. In the case of super antigens, it can be any T cell. So all of the T cells that will be activated will all produce cytokines. So for you to easily remember that super antigens serve as a clamp or glue between the MHC and the T cell receptor, just always look at this picture, super antigen. Typical antigen on the left will only activate specific T cell. Super antigens will lead to non-specific T cell activation because it can just randomly clamp the MHC class II of the macrophage and the T cell receptor of any T cell. That's why it's very obvious in the picture. You have there several types of T cells being activated by the super antigen. So these super antigens, even if at very low concentrations, they can cause the T cells to release large amounts of cytokines. I'm getting carried away with my explanation. So there's massive T cell activation and release of large amounts of cytokines. Indicated in the PPT are the different types of bacterial exotoxins that can be super antigens. Good example is the toxic shock syndrome toxin of Staphylococcus aureus. Toxic shock syndrome toxin is common among women who are using tampons. In the past, women are using tampons instead of napkins. They will insert these tampons into their vaginal canal so that it can absorb the menstrual contents. The problem is tampons can be contaminated with Staphylococcus aureus, and this Staphylococcus aureus can produce toxic shock syndrome toxin, which is a bacterial exotoxin and a super antigen. That's why women who used the contaminated tampons developed organ damage and hypotension secondary to the large amounts of cytokines brought about by the super antigen. Another group of antigen is called the mitogen. This word is derived from antigens that can cause the T cells and B cells to undergo mitosis. An immunogen, the first triangle, if introduced to the immune system, will end up generating immune responses. A mitogen, if presented to T cell and B cell, will result to the proliferation of T cells and B cells in the process called mitosis. Thus, they are called mitogens. So if you want to increase the number of T cells and B cells in the laboratory, you can use these mitogens. These are the mitogens of T cells: concanavalin A, phytohemagglutinin, and pokeweed seed. I'm not looking at the PowerPoint slide, and that is to tell you that you should memorize it because I did memorize it. So if you have a lymphocyte and you want to know whether it is T or B, you can mix this lymphocyte with concanavalin A. If it will multiply in number, then that's your clue. You're looking at a T cell. These are the mitogens of B cells. You have the lipopolysaccharides, you have the lectin, and the pokeweed seed. So the pokeweed seed is a mitogen of both T cell and B cell. Antigens can also be classified as T-dependent or T-independent. When you say T-dependent antigens, these are antigens that would require the help of T helper cells to activate B cells to become plasma cells and produce antibodies. The antibodies will never be produced without the help of the T cells. So these are the antigens processed by the macrophage, presented to the T helper cells, which caused the T helper cells to be activated, and later on, the activated T helper cells will make the B cells become plasma cells and produce antibodies. Please take note of this. There are 5 types of antibodies, but the first to be produced in an infection is IgM. Later on, this IgM will be slowly replaced by IgG and IgA, and by the other 2 antibodies. And the shifting from producing IgM to IgG, IgA, and the other types of antibodies is called class switching. Do you know what causes the B cell to do the class switching? The T cell. Please always remember this. If we are dealing with T-dependent antigens, the T cells play an active role. So, if the immune response is directed towards a T-dependent antigen, expect that you could have several types of antibodies against that particular antigen because class switching is possible because T cell is involved. As mentioned a while ago, T-dependent antigens need the help of T helper before the B cells will be activated to form the antibody-producing plasma cells. And these are the characteristics of T-dependent antigens. They are proteins by nature. Proteins are made up of 21 amino acids, right? So that's why I expect that these T-dependent antigens have different kinds of determinants because proteins are chemically complex. And according to the PPT, T-dependent antigens can stimulate the production of different types of antibodies because there is class switching because of the help of T helper cells. And expect that there is immune memory. T-independent antigens, on the other hand, are most of the time carbohydrates or polysaccharides. And you know very well that polysaccharides are made up of repeating subunits of glucose, galactose, fructose, glucose, galactose, fructose. That's why if you're going to take a look at that microorganism in the PPT, you can see repeating determinants or epitopes. Please go back to the previous slide, the one that talked about T-dependent antigens. Look at the microorganism there. You will appreciate that there are several types of determinants because T-dependent antigens are proteins by nature and proteins are chemically complex. T-independent antigens, on the other hand, are polysaccharide. They have less number of determinants compared to T-dependent antigens. And they are named T-independent because they don't need the T helper cells to activate the B cells to become plasma cells and produce antibodies. So how do you think these T-independent antigens can activate B cells without the help of T cell? They will just simply what? Since they have repeating subunits or epitopes, these epitopes will bind to the surface immunoglobulins present in B cell. And what these repeating subunits or epitopes will do, they will cross-link the B cell receptors. And the cross-linking, just like what is shown in the picture, will be enough to activate that B cell without the help of T cell to become plasma cell. And that plasma cell now will produce IgM. Doc, why only IgM? Because class switching is not possible with T-independent antigens because they did not activate T helper cells, which are the ones that will instruct the B cell to do class switching. And according to the PPT, this type of antigens do not induce cell-mediated immunity because cell-mediated immunity is facilitated, mediated by T cells. And there is no immunologic memory if we're dealing with T-independent antigens. So I hope before you proceed to the next slide, please do memorize the differences between T-dependent and T-independent antigens. So T-independent antigens will activate the B cell even without the help of the T cell by cross-linking the surface immunoglobulins. They can do this because they have repeating epitopes or subunits because they are polysaccharides by nature. So as you can see, the activated B cell will become plasma cell and the plasma cell will only particularly produce IgM. Adjuvant are substances that can enhance the immunogenicity of a particular substance. And most of the time, these adjuvants are added into the vaccines so that they will make the components of the vaccine more immunogenic, thereby making the vaccine more effective. One of the mechanisms on how adjuvants can increase the immunogenicity of the components of the vaccine is through formation of a depot. When you say formation of a depot, the adjuvant will temporarily trap antigens along the site where the vaccine was introduced, and it will only allow slow release of antigens. So if the antigens are slowly released, then the adjuvant will make the immune system react every now and then to the presence of the antigen. And you know very well that in adaptive immunity, the more that you are exposed to the antigen, the higher the magnitude of the response. So take a look at the picture of this adjuvant. Can you appreciate that it's trapping the antigen, only allowing slow release? That is to enhance the magnitude of immune response that we generate against the components of the vaccine. Other mechanisms on how the adjuvant can increase the immunogenicity of the components of the vaccine are, number one, they can increase the phagocytic activity of the APCs. When they do, they can also increase the activity of antigen presentation. So, the higher the activity of antigen presentation, the more T cells will be activated. And with that, the T cells could be producing higher amounts of cytokines. And with the higher amounts of cytokines, you will have higher numbers of B cells that will become plasma cells. And when you have higher amounts of plasma cells, expect that you will produce higher amounts of antibodies. The last topic is all about the pathogen-associated molecular patterns, and these are very important in helping our phagocytes recognize what to eat and what not to eat. Pathogen-associated molecular pattern is defined as any substances shared by microorganisms, and these substances are not present in the host cells. So if they are detected by the phagocytes, that would only mean that the cell in front of that phagocyte is foreign, non-self, and that phagocyte is supposed to engulf them and kill them in the process called phagocytosis. And for the phagocytes to be able to recognize the presence of PAMPs, or the pathogen-associated molecular pattern, these phagocytes should be provided with pattern recognition receptors, the green-colored figures on the picture. These pattern recognition receptors will help the phagocyte detect the presence of PAMPs, pathogen-associated molecular patterns. The most popular pattern recognition receptors are the toll-like receptors, or TLR. Take note that there are toll-like receptors displayed on the membrane of the phagocytes, and there are also toll-like receptors present inside the cells. So the ones displayed on the surface or the cell membrane are the ones that are supposed to recognize extracellular antigens. So just look at the picture and help me identify the different toll-like receptors that are displayed on the cell surface. You have T helper, toll-like receptor 1, toll-like receptor 2, toll-like receptor 4, toll-like receptor 5, toll-like receptor 6. So it's from 1 to 6 except number 3. While those that are found inside the cell, so they are supposed to identify intracellular antigens, especially viruses. You have T helper, toll-like receptor 3, toll-like receptor 7, 8, and 9. So, displayed on the surface, 1 to 6 except 3. And then 3, 7, 8, 9, those that are found inside. These are the different toll-like receptors present on the cell surfaces or the surface of phagocytes. But before I will discuss them one by one, I want everybody to take note that the following PAMPs, or pathogen-associated molecular patterns, are associated with, okay, now let's start with number 1. Lipoprotein, it would always be associated with Mycobacterium species. Lipoteichoic acid, peptidoglycan, these are components of gram-positive bacterial cell wall. Please do remember this. Lipopolysaccharide is present in gram-negative bacterial cell wall. And then zymosan is part of the fungal cell wall. So that's why, let's say for example, for toll-like receptor 1, it is a pattern recognition receptor that identifies lipoprotein as pump. And since lipoprotein is associated with Mycobacterium, then toll-like receptor is used by phagocytes to identify Mycobacterium. So let's go with toll-like receptor 2. You have there peptidoglycan, automatic gram-positive, lipoprotein, automatic Mycobacterium, zymosan, yeast cells. So this toll-like receptor will help the phagocyte identify if the organism in front of it is Mycobacterium, a gram-positive bacterium, or a yeast cell. Toll-like receptor 4 identifies lipopolysaccharide as pump, and lipopolysaccharide is seen in gram-negative bacteria. Toll-like receptor 5 identifies flagellin, a component of the flagella of bacteria. That's why any bacteria, may it be gram-positive or gram-negative, as long as it has flagella, it will be recognized using toll-like receptor 5. Toll-like receptor 6 has similar set of organisms that it can be used to identify gram-positive, Mycobacterium, and yeast cells, because it can identify the following pumps: lipoteichoic acid, which is present in gram-positive bacterial cell wall; lipoprotein again, with present in Mycobacterium; then zymosan, present in yeast cells. So please do memorize the corresponding pumps of the following pattern recognition receptors. So these are the different toll-like receptors that are found inside the cell, particularly in the endosome. And these are the toll-like receptors that will give the cell the idea that there is the presence of an intracellular organism inside it. Most of the time, these intracellular organisms are viruses. That's why, as you can see in the picture, all of the toll-like receptors that are found inside the cell are basically there to detect the presence of viruses. Let's start with toll-like receptor 3. This one detects double-stranded RNA virus. Please always remember, RNA is always single-stranded. So expect that majority of the RNA viruses that can make us get sick are single-stranded, except for one, and that is reovirus. So reovirus, or the double-stranded RNA, will be detected by toll-like receptor 3. If this toll-like receptor 3 is activated, then the cell will know that there is a virus inside it. Okay, reovirus is the most common cause of diarrhea, viral gastroenteritis among children. Next, toll-like receptor 7 and 8. Okay, these are used to detect the presence of the rest of the RNA viruses that are pathogenic to humans, because majority of them are single-stranded. Now, there are also DNA viruses. So, the cell should also have a means of knowing that there are DNA viruses inside it. So, the cell will use toll-like receptor 9. But, there are also bacteria that are intracellular. So, the cell will also use the same toll-like receptor 9 to know whether there are double-stranded RNA viruses inside it and bacteria. And that ends my lecture on antigens. I hope you have learned a lot of things about this chapter so that you will be able to pass our quiz and exam. Thank you very much for listening.